So welcome to our B01 Data Management course on FAIR data. So as you all know, FAIR stands for findable, accessible, interoperable, and reusable. But this is challenging, and it kind of really traverses different research communities, and it requires efforts. And what I really want to get across to you is that if you make your data findable, that's good, and you continue on to the other parts of accessibility, interoperability, and reusability, that's great as well. But you don't have to achieve all of them. That if you go through this step by step and gradually try to achieve parts of FAIR, that can be acceptable in depending on your particular context. And what we're trying to get across is that if you think about how to break these down, how you're trying to do it, that findable can be translated into ensuring that your data has a data dictionary and has unique IDs and is in a relevant publicly accessible database. Accessible, that means that people can go to that database and retrieve your data set easily and using these unique IDs. And if you think about any kind of NCBI, GenBank, or whatever, you routinely might go to that database or download a data set, and that means that data set is accessible. Typically, that format, when you download that data, it will be in an interoperable format that you can use for your analysis, and you should be able to access the metadata as well as the actual underlying data itself. And the metadata should have a defined vocabulary that's relevant for your area. And then if you're able to reuse that, that's great. And if the metadata is sufficiently rich, it might be very useful for your analysis. Sometimes the metadata is lacking, and sometimes there's no getting around that. So that's kind of where we want to be in terms of our own data sets. So how do we approach this and break this down even further? So assign identifiers to our data sets that are universal and unique and shared with our collaborators or the people that work in the lab, and are not specific to our own institute, or these are universal. The data are described in detail with a rich metadata data. That's an established vocabulary. And that these are searchable. For accessible data, we should help to use our identifier and access that data. And this should be open and free and not in some sort of obscure database. For certain data sets, there may be an authentication and authorization procedure, depending on the type. And we should ensure that the metadata is also accessible too. Interoperable. Well, we ensure that the data and metadata are in a format that is easily understood by researchers in a particular area. And that would mean using standard vocabularies that are relevant for our area, and that where relevant our data or metadata should link to other references or other data sets that are within that data set. So for example, your data might be a single sample from your data set might be part of your collection, and that should be referenced inside that metadata. For reusability, then we want rich metadata wherever possible, and that should be have a licensing, so we know that it's reusable for public use. That detailed provenance, that the metadata is very, very detailed, and in that it meets community standards, and therefore the people that you're working with in the wider community can understand how to do that as well. There's a website, fairsharing.org, and kind of has more detail on this. That contains information on databases, policies, and standards, because they're very diverse data sets that you might be dealing with. For example, there's Genomic Standards Consortium, there's the UKRI Open Access Policy as well. There's the BBC Data Sharing Policy, and there's also others such as the Research Data Alliance and the Global Alliance for Genomics and Health. So in terms of a project, so you might see, I don't know if you've heard the term verification, but making data fair needs some thought. So if we think about the project, how we break down the steps, we might get the data and model the domain, which means what types of data do we have? For example, is it gene sequence data? Do we have image data? And therefore we've got identifiers associated with those particular data types. We apply the data standards, so that's labeling the metadata appropriately, using appropriate ontology to describe the data, and make sure the data can be transformed for interoperability into other data sets. So for example, for sequencing data, initially it is not created as a FASTQ file as such. It's a different format, either BCL or typically FAST5, for example, or other types. But FASTQ is the standard way in which data can be uploaded as primary data onto different databases. So therefore, the original sequencing files we've converted into FASTQ, and that would be the interoperable format. There are other ways of sharing data in genomics, for example, as well, and these are called SAM, BAM, CRAM, other files, which can also be shared, but they're not necessarily the standard way of doing that. So then you want your data to be hosted. So this might be using an appropriate database that's relevant for your database, where you submit the data and metadata, and you can check that data is actually accessible. So that's kind of our summary for this. That findable means you can cite it using an accession number. That is accessible means that you can count, so how many files are present, how many samples are present, what's the structure of the metadata. Interoperable means that's interpretable by your people outside your lab, and reusability will mean that it is particularly is reproducible. So unique IDs, the metrics are known, that it's, the future users will understand it, and then it's well documented as well. So that's kind of our summary of this area of FAIR guidelines. Please see the next video on open science.